Right, you can also add the content onto the page while I'm explaining, right? So we have to go step by step to understand the light dependent reaction of photosynthesis. The light dependent reaction, and then under the same topic there is a subtopic, non-cyclic photophosphorylation. Right, first we try to understand what is the meaning of this light dependent reaction, and what is the meaning of non-cyclic photophosphorylation. Right, the light is required for the reaction. That means the energy trapped in solar light, sunlight, is used for this reaction. And what is use this energy? This energy is used for the photophosphorylation. And what is photophosphorylation? The phosphorylation is as we discussed before about the ATP molecule. So ATP is formed by combination of adenosine diphosphate molecule and the phosphate group. So if the phosphate group join to make ATP molecule, then energy is also added to make this ATP molecule, right? So then here the energy is added, and where does this energy come? This energy comes from the sunlight. That's why it is named as a photophosphorylation. The energy of the sunlight, that's called a photon. Usually the light energies come as a photon. This energy added onto the ADP molecule, and then phosphate group is joined to form ATP molecule. So at the end of the light dependent reaction, we're going to learn how exactly ATP are formed. But first of all, you should understand it is a process, not just a single reaction. So where does this happen? The place is a thylakoid membrane. So it is happen at the thylakoid membrane, right? Okay, so what I'm going to add a thylakoid membrane. So I use a box or the long rectangle. This represents part of the thylakoid membrane. Right, to a little bit give the value for this membrane, I can still add a phospholipid bilayer. This shows it's a part of the thylakoid membrane. Then next thing I'm going to add, as we discussed before, the thylakoid membrane just if you recall the basic structure of the cell membrane, it is made out of phospholipid, and proteins are embedded at many different places. So last week we learned about the photosystem. Photosystem is collection of proteins and primary and accessory pigments. Right now I'm going to add one photosystem here. Okay, this represents one photosystem, and I'm going to also add the second photosystem at this place. Right. Last week we learned there are two types of photosystems: photosystem 1 and photosystem 2. Photosystem 1 is also known as 700 nanometer PS 700, and photosystem 2 is known as PS 680. So this photosystem consists of pigments and proteins. And we also learn photosystem are specialized structures. They can absorb energy of the sunlight, and this energy of sunlight moves towards the basically chlorophyll molecules. At the end, chlorophyll molecules get this light energy, and the chlorophyll molecules loaded with this energy from the sunlight, right? So let's start from this photosystem, right? Okay, before that I'm going to also add another group of proteins, 1, 2, 3. So they are protein complexes which is also located inside the membrane, and here also 1, 2, 3. Again 3 protein complexes, because I cannot label everything, otherwise it's so crowded. This at the end this will be so crowded, right? But I can still add this one, photosystem 2. Here I show photosystem 1. Okay, then there is another structure which is also located in the membrane, and this one is a complex. It's a enzyme complex. So this enzyme complex has very specific structure, just like a small motor. It just appears like a small motor, and this enzyme complex is called ATP synthase. These proteins located in the thylakoid membranes, they are not important to learn. What are their names and how their structures are not important, and it is not necessary. Otherwise they have a special name: plastoquinone, cytochrome and plastocyanine, ferredoxin, ferrous sulfate, and NADPH dehydrogenase. But these names are not important. Only you just remember they are proteins, and the role is electron carriers. They can transport electrons. They can take electrons and they can transport. Right. I think we have all the components now to start the reaction. The reaction starts when the light energy is absorbed by the photosystem. So I add this one, these two arrows, and label 1 starting point. So starting point is light energy is absorbed, right? If you look at the notes, yeah, number 1, photosystem 2 absorbs light and energy. That means energy absorb, energy come as a light, absorb, which causes the P68 molecule to excite its electron and pass on to electron acceptors, right? So number one point is explaining exactly the event take place at the photosystem 1. Energy is absorbed by the photosystem 2. When the energy is absorbed, the light energy finally move into the atoms of this photosystem 2. So these the chlorophyll molecules also consist of carbon, hydrogen, oxygen, nitrogen, magnesium, these atoms. So chlorophyll atoms take this energy, and then this energy leads to excitation of electrons. So electrons are going to the higher level. When it moves to the lower level to higher level, and some of the electrons are released from this atom, right? So I represent this arrow showing the electron is released from the atom. Now when the electron is released, I just represent the yellow circle here to show this electron have energy. Electron have energy because the sunlight energy now transfer to the electrons. As we know, the energy cannot destroy, energy cannot create, we can only transfer. So energy transfer to electrons. Okay, now there is a issue. One issue is if the atom lost electrons, atom are unstable, because they are very stable when they have the right amount of electrons. If the electron loss, they have an imbalance because the positive and negative charges are not really balanced. The second thing is these free electrons cannot exist as free electrons because they also unstable. Electron also stable when they move into the atom. But what happened? The light energy caused this event to happen. Then electrons are excited, which left the atoms which are unstable, and then electron is now not free. Okay, then we're going to see how these electrons, the free electrons, moving through these complexes, right? So these proteins are electron acceptors. So they can accept this electron, and then next complex accept the electrons, and then next complex accept the electrons. So when it released from the third complex, this electron lost energy. So there's no circle around it. It lost the energy. So then we have to look where is this energy, to which level they have used this energy, or how energy transfer. So electron energy transfer to another place, then that place has a molecule or anything that has the energy. Okay, simply it's like this. This energy is used to pump hydrogen ion. This side is a stroma. Stroma is the out of this thylakoid, the space out of the thylakoid, and it pump hydrogen ion from the stroma to the inside of the thylakoid. Right now, this energy transfer to the hydrogen or protons or hydrogen ions. So we can understand the energies transfer to hydrogen ion because it used the energy to pump in, and then energy is now with the hydrogen ions. Okay, so still our question is not well addressed because the electron is still free here at this set. And then the next thing is how these atoms in photosystem 2 get stabilized. Right now we are coming to another important thing. That event is not so clear, I mean not yet discovered, but it is something like this. The photosystem, some of the enzyme complexes get activated. So these enzyme complexes get activated, and perhaps some of the energy from sunlight is also important for this reaction. And then there's another reaction take place. This reaction is water molecules split into electron, hydrogen ion, and oxygen. So there's two water molecules important for this one. So two water molecules split to form one molecule of oxygen and electron and protons or hydrogen ions. So this one is not so clear because not yet discovered everything, but it is somehow the enzyme complexes activation, and also maybe the energy from the photosystem too helps to break this water molecule, and the process is called photolysis. The process is called photolysis. Right, I think I hope that you also copy this one, because my explanation with my explanation about this light dependent reaction, you have enough time, so you can add when I'm explaining the process, right? Going back to the notes, the second event, let's see what is it written. Okay, the second point is this creates an electron deficiency in PS2. So this creates an electron deficiency in PS2. So okay, and after that, the third point, this deficiency is filled by a molecule called Z protein, a molecule containing manganese. This enzyme is somehow stimulated by the loss of electron in PS2 to split two molecules of water and photolysis. So second and third point explain that's Z protein, probably the enzyme complex introduced with the Z proteins. This Z protein is activated by the PS2 electron loss, and then two water molecules split to form electron, the hydrogen ions, and oxygen. Right, then the electrons, fourth points, the electron from this reaction are then released to the waiting electron hungry photosystem 2. Right, then this electron can move again back to this photosystem 2. So that means electron from the water molecule going back to photosystem 2. So this electron can fill the gap created by the absorption of sunlight. So atoms need the electrons, and the electrons from the water molecule is going back to a PS2 complex. So note section, it says 4 and 5. 3, 4, and oxygen is released. Right. So then I label this one 3, 4, and then oxygen is released is the fifth point. So oxygen is a byproduct of the reaction, and oxygen is leaving from this thylakoid and also leaving from the leaf. Right, now we found a solution for the first problem, how the electrons coming back to the electron hungry atoms in the photosystem 2. Okay, next we are going to the photosystem 1. So photosystem 1, again light is absorbed, and same thing happen. The light energy moves to the photosystem 1, and this light energy cause excitation of electrons from the atoms. As a result, again electrons are released, and this electron has energy. The round around the electrons in this diagram shows the electron has energy. It's just same as the photosystem 1. Okay, then next there is a subtopic in the notes, formation of NADPH and ATP. Point 6 and 7, it says the electrons are passing through these complexes, electron acceptors. Right now, point 8 says the electron is then passed to electron deficient PS1. The names are not important. That means the complexes, like each complex has a name. Name is given just like additional information, or for your interest to learn a little bit more. But it is not that important for the exam to know this name of these complexes, like a plastoquinone, cytochrome C, and plastocyanil, like names are given. Now this third complex, the electrons which free of energy move into the photosystem 1. I think it's point 8 in the note section says electron is going to the PS1. Now PS1, there is no photolysis because electrons are coming from PS2. It's going to the PS1. There is no photolysis. Right now, let's see what is happened to these electrons released from PS1. Again, these electron acceptor molecules, they take this electron, and it is going from one complex to another complex and to another complex. Right now, these electrons do not use its energy to pump hydrogen ion. Look at here. So when the electron is moving, it uses its energy to pump hydrogen ion from stroma to the thylakoid, inside the thylakoid. But here it doesn't happen. So these are the differences we can see PS2 and PS1. No photolysis, no pumping of hydrogen ion from the stroma. That means the electron still do not lose its energy. So the electron moving out, it has energy. Now these electrons, this time is going to a different molecule, not the complex. They are going to different molecules. This molecule is called a nicotinamide adenine dinucleotide phosphate, NADP, nicotinamide adenine dinucleotide phosphate. Now these electrons together with hydrogen ion, it form a molecule NADPH. So the NADPH has energy now. This energy is now transferred to this NADPH molecule. Okay, what is this NADPH? Right, it is a kind of a molecule considered as a coenzyme, and their role is they are carrying the electrons. They are carrying the electrons when they exist as a oxidized form, and then it become reduced at this level. So you have learned ATP is a universal energy currency that can give energy for any kind of a reaction. You can remember there is another molecules existing similar to ATP. They can also take energy, and this energy transfer to at another point to the reactions, but their significance is not that great as ATP. ATP is a universal energy and their significance is all over. But other molecules like NADPH, they are very specific. That means the NADPH is specific only for photosynthesis. At this level, it is only for photosynthesis. Maybe we can find another place in biological reaction NADPH is involved. But it's a very specific, only 1 or 2, 3, not much. But ATP is energy currency for almost all reactions. Then the breaking and making of molecules take place in the presence of ATP. But here we can understand energy is transferred to now NADPH. Right. Next, we're going to see what is the role of ATP synthase now, because at the moment all complexes done their job. What is left is ATP synthase complex. Right now, where is the energy from this first electron? It is with the hydrogen ion. So these hydrogen ions accumulate inside the thylakoid, and the energy is now loaded to these hydrogen ions. So it creates energy gradient. That means stroma has less energy from this the thylakoid membrane. Outside is less energy and inside is higher energy. So it's an electrochemical gradient. This creates electrochemical gradient. But the energy imbalance is very high, because now it is coming to the maximum level. The energy transfer is very high to this because of the hydrogen ions. There should be a way of transfer this energy to another molecules. So this hydrogen ion can only move through this ATP synthase complex. It can't move through other complexes because it's a specific only for the hydrogen ion. So when hydrogen ion passing through this ATP synthase, there is a complex concept existing, that this ATP synthase molecule just work as a motor, and it's like a sort of almost rotating some of this component, and it can harbor this energy, and this energy can also transfer to make a ATP molecule. That means ADP plus phosphate group, it can create ATP molecules. So hydrogen is now leaving out. So then it can again establish the normal hydrogen ion gradient when this hydrogen ion moving to the stroma. Right, now just to wind up the light dependent reaction, what are the products of light dependent reaction? There are 3 products. One is a ATP molecule. Let's say I make a box. One is ATP molecule. Second one is a NADPH molecule, and the other one is a oxygen molecule. Somehow oxygen is releasing from the cells, and they are not used by the the system or the plants. So they can only use these energy molecules. So ATP has energy, the NADPH has energy. Okay, just to summarize the whole event again, starting point is a PS2. Absorb light, excite electrons. Electrons are accepted by the membrane proteins. Then electron is leaving through these electron acceptors. The energy is used to pump hydrogen ion, and then water molecule split called photolysis. Then the electrons move into the PS2. PS2 again stabilized, and the free electrons can move to the photosystem 1 because it also loses electron when it absorbs light. Then here PS1 stabilized by the electron coming from PS2, and then this electron is moving again to the electron acceptor to make NADPH. Finally, accumulation of large amount of hydrogen ion move through this ATP synthase complex. This create ATP molecule. This is called photophosphorylation. Okay, I think you have to join again because we have done the light dependent reaction and we have completed our first session. In few seconds you can join again once we automatically leave from this session, right? Alright, if you have any question you can post, because I know a bit complicated this light dependent reaction. If you don't get anything, please let me know. Then I can explain again if we miss something by explaining this light dependent reaction. And I also hope you copy this down onto the notes, and let's add the other points on the note sections to complete the notes. So we have stopped at 8 point. So let's see what are the other points.